Okay, so I'm Göran Forsberg. I'm CEO of Cantargia. Nice to be here and present. I guess most of you are familiar with Cantargia. We are an antibody company based in Lund in South Sweden. We are in phase 2A clinical trials in non-small cell lung cancer and pancreatic cancer. We are very much focusing around one molecular target called IL-1RAP, which is a target with several opportunities both in cancer as well as in autoimmune diseases. And we'll go through that in much more detail. What's extremely exciting for us as a company is that we actually have granted IP on this molecular target, which is valid until 2032, which gives us an extra layer of protection in addition to our composition of matter patents on lead compounds, which are valid until 2035. We've been, let's say, working very hard to get a strong management team in place who has already done this type of development beforehand from all aspects of the product, from production until clinical development. We are listed on this main market here in Stockholm. We have almost 6,000 shareholders right now, and our market cap is about $130 million. We have very strong investors backing us. Sunstone, a Danish VC, is a main investor. But besides that, we have 4 different pension funds, Swedish, Alecta, and 3 AP funds. We have a fund called Nordic Cross, part of Carnegie Asset Management, and we have Handelsbanken funds. Our pipeline looks like this. CAN04 is our main project. It's in phase 2A for non-small cell lung cancer and pancreatic cancer. It's a quite complex trial. We'll go through that in more detail later on. But there are also lots of opportunities to expand CAN04 into several other cancer indications. Leukemia is one where we have presented lots of data, but there is actually much more, and I will show that as well. CANXX is our second project. It is, again, antibodies against the same molecular target, IL-1RAP, but with completely different properties. And they are in development for autoimmune inflammatory diseases. So IL-1RAP is a target involved in inflammation, and what is the link between inflammation and tumors? So to get a cancer to grow in the body, you need 2 independent events to happen. First of all, you need mutated cells, and we all know that. But secondly, we actually need some kind of tumor-promoting inflammation. And this is really giving some kind of tumor microenvironment where the early cancer cells can grow in and not be eradicated from our own immune system. And what the inflammation will lead to is immune suppression around the cancer cells. It will help the primary tumor to metastasize, and it will also generate an environment which leads to therapy resistance, both chemotherapy as well as immune therapy. And our idea is that we can really block all these events with our therapy. And we got a great favor from Novartis, who made a major trial in cardiovascular disease and got a side effect which they probably didn't think about, but the side effect was that they reduced the risk of getting lung cancer. And this was a trial in more than 10,000 patients, so it was a huge clinical trial. The patients were followed up to 6 years. And what we showed was that by treating inflammation in a similar way as we do, and I'll explain the differences in a second, they could reduce the risk of lung cancer with 67% and the risk of dying in lung cancer with 77%. And this is the interleukin-1 pathway, where we block the receptor. And what's very obvious here is that you have a dose response, which makes the data even more credible. Novartis has then changed their program and basically stopped the development in cardiovascular disease and turned this into a cancer drug. So now canakinumab is in 3 different phase 3 trials in non-small cell lung cancer in various stages of the disease. And there are lots of phase 1 and phase 2 trials in other cancer forms, in renal cell cancer, gastroesophagus, colorectal, as well as other lung cancer trials. And then we need to understand why our compound has potential advantages compared to Novartis. So the interleukin-1 system, and it's difficult to see here, so I do it like this, consists of two different forms of interleukin-1. You have alpha, which is found predominantly on different cells in the tumor tissue, and you have interleukin-1 beta, which is a soluble cytokine. And Novartis antibody is directed against IL-1 beta, which means that they can remove that from the blood, but the IL-1 alpha in the tumor is still signaling. What our antibody is doing is binding the receptor IL-1 RAP in a way that it's blocking both interleukin-1 alpha and interleukin-1 beta signaling. And that's one part of the mechanism. But since we have an antibody which is binding to the tumor cells, we also have something called ADCC, which means that the tail of this antibody will bind to natural killer cells and macrophages, which will then come into the tumor and eradicate IL-1 RAP expressing cells. So we very clearly have a double mechanism, which Novartis doesn't have. So, yeah. So now we have an ongoing phase 2 trial. The phase 1 data were presented in an oral session at ASCO earlier this year, and was very encouraging. At that point in time, we showed that we have a very good safety, but we have a very clear, pronounced, quick effect on relevant biomarkers in the tumors, which are, let's say, bad news for the patients, like interleukin-6 and CRP. We didn't get any responses, which is unfortunately very difficult to get in late-stage patient populations in cancer, and especially in pancreatic and non-small cell. But we got lots of stable diseases. But what we're doing right now is that we have expanded the trial into 3 different arms. So we have one monotherapy arm, which is very important to get more safety data, to get more biomarker data. But from a commercial point of view, we believe very much in combination therapies, in combination with chemotherapy in both non-small cell lung cancer and in pancreatic cancer. In non-small cell lung cancer, we do a combination with gemcitabine cisplatin, and in pancreatic cancer, we do a combination with gemcitabine Abraxane. So it's typically first-line chemotherapy. So we really want to get patients on board as early as possible. We are currently recruiting. We will expect to get data from a combination therapies in 2020, early 2020. And at that point in time, we will select which of the indications, non-small cell lung cancer and pancreatic cancer, that looks most promising, and include another 30 to 50 patients in that patient population, perhaps even use biomarkers to select the most promising subgroup of patients. So then just looking into IL-1 RAP, approximately 80% of the patients with non-small cell lung cancer have IL-1 RAP on their cancer cells. It's about 70% of pancreatic cancer patients. But as you can see here, there's a large number of both solid tumors as well as leukemia, where the majority of patients have IL-1 RAP. But what's interesting is that all patients have IL-1 RAP in their tumor microenvironment through various immune suppressive cells like myeloid-derived suppressor cells. So the opportunity is really to treat all patients, even if they may not have IL-1 RAP on the tumor cells. So to summarize this, our antibodies are really designed to attack cancer cells, to attack metastatic cells, and to attack tumor-promoting immune cells to get them eradicated. In 2018, we did some very exciting discoveries. So first of all, we could actually show that we have anti-metastatic effects. But what was also very exciting was that we could show that we have synergistic effects with chemotherapy. So as you probably know, chemotherapy is great initially. You often get tumor shrinkage, but very quickly the cells get resistant to chemotherapy, and then the situation gets worse and you get metastasis and you get growth of the original tumors. What we could see is that when we combine chemotherapy with our drug, not only metastasis stop, but we also get continuous shrinkage and much better responses. And this is just one example where we have been looking into a very aggressive model of non-small cell lung cancer, where we can treat the mice with one of the standard therapies, which is cisplatin, which is used for non-small cell lung cancer. It's not working in this model. You can use gemcitabine where you get some effect. Patients often get a combination of cisplatin and gemcitabine, which has some effect here. But when we add our drug, it's really, really enhancing the therapy. And even more interesting is that we notice that we get less side effects from a chemotherapy when we have an anti-inflammatory drug at the same time, which has to do with cachexia being the side effect of chemotherapy. And cachexia is driven by IL-6, which if you remember, we have a very good effect on. So by reducing IL-6, we can also reduce the side effects from chemotherapy. So that was very much our cancer project. And now we're getting into autoimmunity, inflammatory diseases, where IL-1RAP is still the target. And we talked about the IL-1 system, but it's even more complex than that. IL-1RAP is a co-receptor not only for IL-1, but also for IL-33 and IL-36. And if you're following the field, IL-33 and IL-36 has become very, very hot targets for therapy. And for instance, Sanofi presented some very encouraging data on IL-33 blockade in asthma a while ago. For instance, Boehringer Ingelheim presented very strong data in psoriasis with an IL-36 blockade. But what we can do here, if we design the antibodies properly, is to actually have antibodies blocking all three systems at the same time. And very often these IL-1, IL-33 and IL-36 works in, let's say, together, which means that we have unique opportunities to block rescue mechanisms from these other therapies. This is in preclinical development. We have an ongoing collaboration with the US company called Panorama Research Incorporated. We are an antibody engineering company, so they really design the optimal antibodies, and we will then take them into clinical development. And the plan is to get the first clinical candidate in 2019 being selected, and then as rapidly as quickly put into clinical development. So we expect to have a great news flow in the next year and two. So what I didn't talk so much about is that we're actually planning a new clinical trial in our CAN04 project, which is going to be carried out in the US. We are in preparations for the pre-IND meeting with the FDA. And once we have had that pre-IND meeting, we will also be able to communicate more details on the trial design. But I can tell you it's going to be some kind of combination trial in a new setting, which we're not addressing right now. It is obviously phase 2A monotherapy, has gone very quickly to recruit, so we will be able to present data during 2019 from that part. We will probably be able to present a little bit on the clinical progress in the phase 2A combination therapy during 2019. Preclinical progress, we have a huge preclinical program to understand more about our mode of action and exactly which combinations which are useful, so we will present data on that. The new project, CanXX, we have decided to select clinical candidate this year, and we will communicate around that once the IP is ready. And then more about the UNIT's clinical trial once approved. And in 2020, the phase 2A combination results and expansion, if it's going to be non-small cell lung cancer or pancreatic cancer. So I'll finish off with the slide I started with, and I'll be very happy to take any questions if you have.
Thank you.